So as we pick up our second part of our special senses lecture, we will continue with vision. Now we're going to really tie off vision by discussing the specific photoreceptors and exactly how they convert this light, this energy, into neural impulses and how it gets deciphered by our brain. So just to repeat, rods and cones, these are our photoreceptors of the eye. They're located in the retina, and their job is to convert light energy, remember wavelength and amplitude, into neural impulses, action potentials that are going to be propagated down the optic nerve through the optic radiations and then ultimately terminate in the occipital lobe or the primary visual cortex. Now again, these rods and cones were named for the appearance of their outer segments. So we see here the cones are named cones because of their cone shapes, and the rods are named rods because of their rod shape. Rods and cones contain photopigments necessary for the absorption of light that will initiate the events that lead to the production of a receptor potential. Now again, rods contain rhodopsin, cones contain photopsin. We're going to be focusing on the light process in rods. Now again, cones contain 3 different photopigments, one for each of the 3 types. Remember our differentiation of red, green, and blue. Photopigments respond to light in a cyclical process. And this is what we're going to examine in our next slide. So we're going to examine this cyclical process. So let's first take a look at the components of the photopigment. We start with these protein discs. The discs are made up of the photopigment rhodopsin. Now rhodopsin is a dimer. It's made up of 2 chemical components, one retinol and one opsin. So now what we're going to do is we're going to discuss this process here in its entirety, and then we'll take it step by step. Now structurally, rhodopsin is classified as a chromoprotein. What this means is it's derived from the meaning of color. It is made up of opsin, which is a colorless protein, and cis-retinol, which is a pigmented molecule derived from vitamin A, which is why it's so important that we have vitamin A in our diet. Now, when the eye is exposed to light, cis-retinol component of rhodopsin is converted to trans-retinol, resulting in a fundamental change in the configuration of the rhodopsin molecule. And we see that at step 1. Light is coming in, it's striking rhodopsin, and it's isomerizing retinol into a trans or straight type of conformation. As a result, trans-retinol separates from opsin, which is the bleaching. Now, the change in configuration initiates a phototransduction cascade within the rod, where light is converted into an electrical signal that is then transmitted through that optic nerve that we talked about before to the primary visual cortex in the brain, again the occipital lobe. The change in configuration also causes opsin to dissociate from retinol. This is that bleaching that we see. So we're left with this colorless opsin. Bleaching limits the degree to which the rods are stimulated. Ultimately, this decreases their sensitivity to bright light, which allows cones to mediate vision in bright environments. So basically what we're talking about is what happens in light conditions to rods. Now we can continue with step 3. Now the trans-retinol that was released during bleaching is either going to be stored or changed back to cis-retinol, right here with something called retinol isomerase. It's an enzyme that's going to aid in the conversion of trans-retinol back to that bent cis-retinol shape. Once retinol is back in its cis or bent conformation, it's going to be transported back to the rods. Now, this is known as recycling. It's allowing for the regeneration of rhodopsin. Now, rhodopsin regeneration takes place in darkness, and is central to dark adaptation. And then a little later in this lecture, we're going to talk about dark adaptation versus light adaptation. Now, when dark adaptation occurs, when rhodopsin levels depleted from bleaching in a brightly lit environment gradually increase, this enables those rod cells to become increasingly sensitive to dim light. So this is where we are in this state. We allow for cis-retinol to bind back to opsin, forming that colored pigment. And again, we are now being active. We're being sensitive, increasingly sensitive to those dim light conditions. Now again, as long as the stimulus of light is being introduced into the equation, so we're outside, daytime, or there's any type of light, this cycle is going to be held in this photo-bleached or this bleached state until the stimuli or light is removed. Once the light is removed, we will return back to our colored pigment. So this is our cyclical cycle of bleaching and regeneration of rhodopsin. So we're going to elaborate and discuss more this adaptation to dark and light conditions and how these are affected based on that cyclical cycle, that regeneration and bleaching processes that we just saw on our last slide. So we're going to start with light adaptation. So this occurs when an individual moves from dark surroundings to light ones. It takes seconds for our rods and cones to adapt. And the reason being is if we go back to that cycle, when light strikes that photopigment, the bleaching that occurs, the separation of opsin to retinol, is relatively quick. It takes seconds. And it takes seconds for those cones to adapt. Now, during those seconds, what can occur? Sometimes you see glaring, right? You see this bright white glaring. And what that is the overstimulation of the cones and rods from that introduction to light, and until they really account and the cones take over, we see a glare for a few seconds. Now, dark adaptation is completely opposite. This occurs when one moves from a lightened area to a darker one. Now, this is completely different because this can take a long time. It could take several minutes for you to be able to see. Once you move from bright to dark, really all you see is black. Right? Darkness. You really have no depth of view, no perception, and it takes times for your eyes, we say, to adjust, right? So what's happening during this adjusting period? Why does it take so much longer? Well, remember, light struck these rods. We are in a photo-bleached state, right? So we have rhodopsin that has been bleached, and so we have transretinol sitting off somewhere and opsin sitting off somewhere. Now we have to allow them to regenerate. So that regeneration takes time. Regeneration here takes time. So it takes minutes for that opsin and retinol to come back together. And the time for retinol isomerase to convert transretinol back to cis-retinol, and then cis-retinol making its way back over to opsin and then forming that dimer again, it takes minutes, which is why our eyes have to, this in parentheses, adjust to the darkness. So now the final bullet point kind of explains that. Part of this difference is related to the rates of bleaching and regeneration of photopigments in rods and cones. Our next step in this process, now that we have discussed what happens here on step 1 with the photo bleaching and regeneration, we kind of have to put this entire system together, look at the entire bipolar cell and say what happens with the outer layer, the inner layer, and then at the synaptic terminals. Now this gets a little tricky, and the reason why it gets tricky is because it goes contrary to belief. Everything that we've talked about to date is and leads us to believe that when we excite a neuron, it propagates an action potential. So typically on means that the neuron's on, right? If we put it in a simplistic fashion. With these bipolar cells in the retina, it's actually opposite. And we're going to write this out for you, so I want you to keep this in the back of your mind as we discuss this. Off means on, and on means off. So what does that mean? This means we have two types of bipolar cells. We have bipolar cells that are called on bipolar cells, and we have bipolar cells that are called off bipolar cells. So exactly what's occurring here? Now in light strikes this photoreceptor, which causes the photoreceptor to be hyperpolarized. Well, how is it being hyperpolarized? This is due to the activation of opsin, which activates transretinol, giving energy to stimulate a G protein coupled receptor to activate this right here, PDE, phosphodiesterase, which cleaves cGMP into 5-cGMP. Now in photoreceptor cells, there is an abundance of cGMP in dark conditions, keeping cGMP-gated sodium channels open. So when we activate PDE, phosphodiesterase, this diminishes the supply of cGMP. By diminishing the supply of cGMP, we're breaking down the cGMP, we are lowering cGMP concentrations, which essentially closes a lot of our cGMP-gated sodium channels, which means that we are decreasing sodium inflow, which is causing this hyperpolarization event. Now, by causing the hyperpolarization event, we're essentially closing some of our voltage-gated calcium channels, which will reduce the amount of, and we'll go here, the amount of glutamate from being released into the synaptic cleft. Now, glutamate is an inhibitory neurotransmitter. Now, by causing less glutamate to be released, this causes the on bipolar cells to lose inhibition and become active, while the off bipolar cells lose excitation, they become hyperpolarized and become silent. So what are we talking about? We're talking about this. We're talking about keeping the system off by keeping it hyperpolarized, right? So we're going to keep the system off by keeping it hyperpolarized, which essentially is going to turn it on. Okay, so that's what I mean when we say off equals on. Again, this is in light conditions. So let's just reiterate that we're talking about in light conditions. Now again, there's that off equals on scenario, right? That off equals on scenario. Now let's take it to dark conditions. So let's look at our next slide and kind of flip the switch and see what happens. So in darkness, rod photoreceptor cells release the neurotransmitter glutamate. So we're going to highlight glutamate because this is an important inhibitory. So let's highlight inhibits bipolar cells from receiving transmitting signals to ganglionic cells, which provide output from the retina to the brain. So let's write this out again and look at the steps. So what we're saying is, remember in the light we were off, which equals on. Here we are going to be on, which equals off. So let's put our on bipolar cells here, bipolar, and our off bipolar, and now discuss exactly what happens. So let's pick back up with our rhodopsin, so our colored product here. This is opsin and cisretinol. We are in the absence of light, so we do not get any photobleaching, which means that phosphodiesterase is inactive, which allows for a high concentration of cyclic GMP. That cGMP, those high concentrations, allow all these sodium-gated channels to open, flooding in a large amount of sodium, allowing for depolarization. Now, by allowing for that depolarization, these calcium channels in the synaptic end bulb are open, and we are flooding glutamate into the synaptic cleft. Okay, so we're flooding glutamate into the synaptic cleft. So what does this mean from a cyclical or process standpoint? Now, by turning and putting glutamate in the synaptic cleft, what we are essentially doing is by hyperpolarizing, we are exciting the on bipolar cells. We are exciting the on bipolar cells. Well, how are we doing that? We're doing that by essentially turning the system off. So on equals off. How are we turning it off? We are releasing this inhibitory neurotransmitter. Okay? So again, we are releasing glutamate, which is going to inhibit the on bipolar cells and excites the off bipolar cells. One of the last things to talk about with regards to vision is the neural pathway. So what happens after rods and cones are converting that light energy into neural signals, and then they're directing them to the optic nerves? So the pathway occurs like this. Once the optic nerve leaves the eyeball, we are converging onto this, essentially an X. If you go back to our brain dissection that we did in AP1, we have this crossing over of these nerves, of cranial nerve 2, the optic nerve, forming the optic chiasm. We then follow these optic tracks to the lateral geniculate nuclei of the thalamus, and then from there these optic radiations allow the information to arrive at the primary visual area of the occipital lobes for perception. We can use a picture to follow this visual pathway. So again, cranial nerve number 2, we'll start with the optic nerve right here. See if we could use a blue, maybe that'll show up better. Yeah, from the optic nerve and the optic chiasm, the actual crossing over event that we see, through the optic tracks, so we'll follow it here. And from the optic tracks, we're going to go to the lateral geniculate nuclei that are located in the thalamus. So see if we can make a little circle right here, which will then carry us through the optic radiations. So these radiations that are going to move to the primary visual cortex of the occipital lobe. Now, unique to humans, and particularly our great vision, or great sense of vision, we have the anterior location of our eyes, which lead to an overlap, right? Our nose is a good example of this. Our nose lies right in the middle of our field of view. How come we don't see this all day long and it's not interrupting our vision? Well, our brain kind of erases this, right, and sees it as a redundancy. Now, this overlap gives us binocular vision. So what we can see if we look here at our left and right eyes, we see that we have this overlapping, right? And we see the orange from the left eye, and then the orange here from the right eye, and the red from the left eye. This overlapping field of view. It's kind of like a Venn diagram where we keep overlapping these circles. Now, the two visual fields of each eye are nasal, medially, and temporally, laterally located. Visual information from the right half of each visual field travels to the left side of the brain. There's that decussation again. So everything from the left eye is really translated on the right side of the brain, and right eye to the left side, vice versa. Visual information from the left half of each visual field travels to the right side of the brain. So let's separate our left and right eye, and we basically see the overlap. We see the temporal half in red from the left eye, and the nasal half from the left eye, and then the overlapping darker yellow, the nasal half from the right eye, and the temporal half from the left eye. Go back 2 slides. When we overlap these, we get our binocular vision. This brings us to our final sense, hearing and equilibrium. Now again, we follow the who, the what, the where, the when, the why, the how. We're talking about the transduction of sound vibrations by the ear's sensory receptors, turning them into electrical signals, and this tends to be 1,000 times faster than the response of light by the eye's photoreceptors. Now we're talking about hearing, right? We're talking about transducing sound vibrations into the brain, translating those sensory receptors into electrical signals. But what we are not yet delving into is that the ear is also going to contain receptors for equilibrium, essentially balance. Now the ear is divided into 3 regions: the external ear, the middle ear, and the inner ear. let's take a look at a picture and kind of outline our external ear, middle ear, and inner ear and some of their associated structures. So we're going to go ahead and start with our external ear, and we'll see that the external ear is pretty much going to be this triangular shaped region from what you can touch, the outside portion of your auricle, your ear, which is made up of the helix and the lobule down here, helix and lobule, which brings us into our external auditory canal. So this region right through here. Now, if you remember, we have specialized glands called ceruminous glands that line this canal cavity and makes up this earwax that protects that canal. And then lastly, we have our tympanic membrane. So this is what's going to take those vibrations of sounds and move them into the fluid of the inner ear. The next structures or region that we're going to look at is the middle ear, and our middle ear will highlight in blue here. The middle ear is going to include a couple of things. Our 3 auditory ossicles, well 6, 3 on each side, our malleus, hammer, our incus, anvil, and our stapes, or stirrup. These bones will vibrate from the sounds coming in the tympanic membrane and move those vibrations into the inner ear. Now, one last structure we're going to talk about is right here, and it's this structure down here. It is the auditory tube, also known as the pharyngotympanic tube, also known as the eustachian tube. Now, notice how this is at a steep 45-degree angle. Interesting note, in children this tends to be more flat and horizontal, which is why when kids get sick they tend to have a higher propensity to have ear infections, because that mucus kind of sits and builds and stays within this canal, within this tube, which causes and leads to infections and bulging of this tympanic membrane. As children grow a little after the age of 2, this tube starts declining and their propensity or predisposition to have ear infections kind of decreases. The next region we're going to look at is the inner ear, and we're going to go ahead and highlight the structures of the inner ear right here, and we'll say, okay, well, what makes up the inner ear? So in here we're going to have a couple of windows, a round window and a oval window, and that oval window is here underneath the stapes. We're going to have, we'll highlight the semicircular canals and the cochlea. Okay, so the semicircular canals, I'll give you a little hint towards what it does, balance, and the cochlea is really for true hearing. Now notice our cranial nerve number 8, our vestibular cochlear nerve, which makes up these 2 branches, the vestibular branch and the cochlear branch. Look, the vestibular branch goes to the vestibule or the semicircular canals for balance, and the cochlear branch goes to the cochlea for true hearing. So let's put to print what we just talked about in our previous slide. The external outer ear contains the auricle or the pinna, the external auditory canal with those ceruminous glands, and the tympanic membrane or the eardrum. The function of the auricle is to capture sound. Now, let's talk about ear size. We're going to digress for a second. Our early hominid ancestors had really, really large ears. Not only were they larger, but they also faced more forwards. What was this for? This essentially captured sound better. We can mimic the ability our ancestors had. If you take your hands, cup them, put them behind your ears, and push your ears forward and have somebody talk or even yourself talk, you'll see that this magnifies the sound. It's able to capture more sound waves and direct it into the ear. This was a great feature to have, especially if we were trying to avoid predation. The external auditory canal is going to help transmit that sound that the ear captures down to the eardrum. The ceruminous glands again secrete earwax to protect the canal and the eardrum. Moving to the middle ear, the middle portion of our ear, we have the 3 auditory ossicles. These are again the smallest bones in the body, the malleus, the incus, and the stapes. Sound vibrations are transmitted from the eardrum through these 3 bones to the oval window. The auditory tube, pharyngotympanic tube, also known as the eustachian tube, extends from the middle ear into the nasopharynx to regulate air pressure in the middle ear. So when you plug your nose and force air and your ears pop, forcing that air up that eustachian tube helps to regulate pressure inside your middle ear. That's why you get that popping sensation when you do that sample, that experiment. So we can use a picture to zoom on into that middle ear, looking at the eustachian tube over here, our auditory ossicles here, our tympanic membrane through here, and our external auditory canal there. We can see how all this kind of builds together and leads to, if we see the stapes here, is leading to the oval window in the cochlea. Lastly, we have the internal ear. This is made up of the cochlea, which in Latin means labyrinth. This is going to translate those vibrations from the tympanic membrane, from those auditory ossicles, into neural impulses that the brain can interpret as sound. And then we have the semicircular canals that work with the cerebellum to decipher and translate and identify for balance and equilibrium. Now, when we talk about balance and equilibrium, we're talking about two different types of forces. The two forces that we're talking about are static and dynamic. So two different types of equilibrium. So we can use a picture to look at the cochlea and the semicircular canals. We're going to get deep into the structures and features of each of these parts and how they work, but I want to point out a few important regions. Number one, let's look at this structure right here. Below the stapes, underneath the stapes, is the oval window. This is the opening into the cochlea that sound waves are going to travel. Now remember, we're traveling through fluid. The fluid inside the cochlea is more like water. It's a watery consistency. It is water with a few dissolved ions in it. But now here's the problem. Water cannot be compressed. So what happens is, as sound waves, vibrations, come into this window, the vibrations are going to travel through the fluid, which causes a displacement of the fluid. That brings us to our round window. Now the round window is super important because the round window is going to bulge as those pressure waves come in. So as the pressure waves come in this way and move through the cochlea, the displacement of water is going to be absorbed and bulged by no more than our friend here at the round window. So these guys are like two thin sheets, two thin membranes that work opposite. As this one bulges in, the oval window bulges in, the round window bulges out, again absorbing the pressure exerted by the sound coming in. Now a couple other things we see the cochlea with the cochlear ducts. Okay, so we go right here, the cochlea with their cochlear ducts within. We have the semicircular canals. This is going to contain the bony labyrinth where the paralymph is going to flow, as well as the membranous labyrinth that's going to contain endolymph. So in the membrane, endo is within, paralymph flowing around that membranous labyrinth. And again, this is going to move a apparatus within the ampulla of the semicircular canal located in the utricle here, in the utricle. And we'll look into that when we do equilibrium. But for now, we're going to focus on sound transduction and how we interpret sound waves into actual sounds. So vibrations are transmitted from the stapes through the oval window to the cochlea as fluid pressure waves are transmitted into the paralymph of the scala vestibuli. Okay, so here's one of our first definitions that we're going to look at, scala vestibuli. So the staircase into the vestibule. From here, pressure waves travel to the scala tympani, so the staircase membrane, and then to the round window, which bulges in the middle ear. Now, let's look at this scala tympani. And we're going to look at it on the next slide as we see exactly what this staircase structure does and how it operates. So we could follow the sound waves in through the oval window as they travel through the paralymph-filled scala vestibuli and scala tympani. Now the two scalas communicate with each other through an opening at the apex of the cochlea. So where is that opening? Right here. That opening is called the helicotrema. Now that opening is going to allow for the wave motion to transmit the endolymph inside, right here, the cochlear duct. We can see where the vestibulocochlear nerve, so if we draw a line, our vestibulocochlear nerve, cranial nerve 8, comes in and branches and moves through the spiral ganglia into the cochlear ducts. Now pressure waves travel from the scala vestibuli to the vestibular membrane to the endolymph of the cochlear duct. This in turn causes the basilar membrane to vibrate. This moves the hair cells of the spiral organ, which is also known as the organ of Corti, against the tectorial membrane. These cells, the movement of these cells, generate nerve impulses in the cochlear nerve fibers, which is translated into actual sound. So we can put it to picture. So sound waves come in, vibrates the basilar membrane, which in turn is going to stimulate the hair cells, or the stereocilia, on these outer and inner hair cells, which will then vibrate, so that will be number 2, which will then vibrate the tectorial membrane, which will send those nerve impulses down and through the vestibulocochlear nerve. Let's talk about these stereocilia for a while and understand how we're able to hear a wide array of sounds. The best analogy I have is a harp, right? So if you know, a harp is made up of all these different length strings. Okay, I know it's not the best picture of a harp, but we know that the shorter strings are for the higher notes and the longer strings are for the lower notes. So the stereocilia here, and if we kind of zoom on in, we see that they are varying lengths. So the stereocilia are built like a harp. The shorter hairs are going to be receptive to the higher notes, whereas the longer hairs are going to be receptive to the lower notes. So to make a sound and to understand a sound, kind of like smell, where we put all those keys together to make a song, well, here's the same thing. Those hairs are going to vibrate depending on the frequency of the sound, and we're going to put these strings, play them together to make music interpretive as hearing. So we can summarize the entire process, putting it all together, starting with 1, the external ear. The sound waves come in, vibrate the tympanic membrane, which in turn vibrate the auditory ossicles, moving those sound waves from air conduction now to water conduction. We are now inside the scala vestibuli. So pressure waves travel from the scala vestibuli. So we're here into the vestibular membrane. Now this is going to carry impulses to the endolymph of the cochlear duct. So we're going from here to here. From the cochlear duct, the basilar membrane, so we're shooting way up here, the basilar membrane is going to start vibrating. It's going to move the hairs of the spiral organ of Corti, which will then move against the tectorial membrane, which will then generate, it's going to allow these cells to generate nerve impulses into the cochlear nerve fibers. So that's the summation, if you have to put it to steps, for the transduction of sound from the external ear all the way to the inner ear. The cochlear nerve fibers from the cochlear branch of the vestibular cochlear nerve, we see that the axon synapse with neurons in the cochlear nuclei in the medulla oblongata. These impulses will then travel to the medial geniculate nucleus of the thalamus and end in the primary auditory area of the cerebral cortex, which is located in the temporal lobe. We could put it to drawing, identifying first order, second order, third order neurons. So that cochlear branch comes into the medulla oblongata. The superior olivary nuclei in the pons are going to move it up to the thalamus. From the thalamus, we're going to use that relay center to relay information to the primary auditory area in the cerebral cortex. Now, true hearing was only half of this equation. The second half is going to be equilibrium, or balance. And as we said before, it exists in two forms. Static equilibrium is number one. This is the body's ability to maintain a static or positional force relative to gravity. So in other words, this is our ability to essentially stand erect, straight up and straight down, and interpret that gravitational force on our body. The second is dynamic equilibrium. So this is the maintenance of the body's position in response to sudden movements, like rotational forces. So your ability to interpret rotational forces on your body. An example would be turning your head left or right. So this is a dynamic force. Or if you're driving on the highway, getting off an exit, and you have this large sweeping exit, that's a rotational force. Or riding on a roller coaster. You know, sitting still at the beginning of the roller coaster is a static force, and then going upside down in a loop-de-loop would be your understanding of that dynamic force. Now, the vestibular apparatus, this is going to have the organs that maintain equilibrium. It includes the saccule and the utricle. Both are otolithic organs. So we say, well, what are otoliths? Otoliths are calcium carbonate crystals. Now, fish have a ton of otoliths. This helps them interpret up, down, left, right when swimming in a medium like water. We don't have as many otoliths as fish, which is why we have a hard time deciphering up, down, left, right when we're put in a liquid like water medium, which is why when people are wakeboarding or surfing, they should wear a life vest, because when they tumble in the water, most people swim to the bottom and drown because they lose the ability to know which way is up or down. Now, lastly, we have our semicircular canals. This is where our endolymph and paralymph is going to flow, essentially affecting the saccule and the utricle. Now, the otolithic membrane sits on top of the macula. Moving the head causes gravity to move it down and over hair cells. The hair cells synapse with neurons in the vestibular branch of the vestibulocochlear nerve, which propagates action potentials. So let's take a look at the utricle and the saccule. Now, what happens? We have our vestibular branches of the vestibulocochlear nerve. They see that these hair cells, kind of like in taste, smell, again, the same situation, these hair cells are being nourished and supported by these columnar supporting cells. And the stereocilia, the hair bundles, are going to be supported and embedded inside this otolithic membrane that has those calcium carbonate, those otoliths, embedded on top. Now, in the next slide, or the two slides after this one, we're going to look at a detailed cut of the ampulla and see exactly what happens. But just to give you a quick clinical connection here, a real life connection here, and what you're feeling when you're on a roller coaster. So a couple things occur here, and this is how information and action potentials are translated. When you're going on that roller coaster and you're clicking up that big hill, so you're going all the way up the top of this roller coaster, and it's click, 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 click. And once you hit that top and you're about ready to go down, you start this massive decline, right? You get a feeling of weightlessness. You're like, wow, how come I'm feeling weightless? Am I in a zero gravity situation? The answer is no. What's happening is that rapid acceleration, that rapid downward force physically takes this otolithic membrane and pulls it off of these hair cells, these stereocilia. And as a result, the brain loses the feeling, the propagation of these action potentials, because the otolithic membrane is no longer moving against those hair cells. So you get this feeling of weightlessness. Now, on the flip side, as you come down, you feel everything sinking into your butt or into your stomach. And then you say, wow, I feel so heavy. Is there something pushing down on me? The answer is no. What's happening is this otolithic membrane is now slamming back down because of that change of acceleration, change of force is slamming back down onto those hair cells, and the action potentials are coming so quickly, the brain interprets it as this extraneous force coming down on your body. Now we could take a detailed look at the two types of hair cells here. We have our otoliths, our calcium carbonate on top, our otolithic membrane, and our two types of hair cells in our hair bundle, kinocilium and stereocilia. So the question is, how do these two work together? So bending the stereocilia towards the kinocilium depolarizes the cell and essentially results in an increased afferent activity. Now, bending the stereocilia in the opposite direction, away from the kinocilium, actually hyperpolarizes the cell and results in a decrease in afferent activity. The semicircular ducts work in pairs to detect head movement with regards to angular rotational acceleration. Let's look at those three bony semicircular canals that are responsible for dynamic equilibrium. Now the ducts lie at right angles to each other, which allows for rotational acceleration or deceleration. Think of it as an X, a Y, and a Z axis. An ampulla in each canal contains the crista with a group of hair cells. Movement of the head affects the endolymph and hair cells ultimately, which generates a potential leading to nerve impulses that travel along the vestibular branch of the vestibular cochlear nerve. So we can put it to picture. Look at the ampulla that contains the cupula that has the hair bundles, hair cells, and supporting cells surrounding. That leads to the ampullary nerve that's going to lead to vestibular cochlear branch. Think of this as a mushroom. As a rotational force, if I spin you around to the right, the fluid is moving this way. This little cupola, I call it a little mushroom because I think it looks like a little mushroom, is going to bend to the right. That bending is going to be deciphered by your brain as a rotational force. Now, we talk about how vision and hearing are intimately connected. Your eyes are going to account for that rotational movement. This involuntary eye movement is called nystagmus. So if I were to take you, spin you around in a chair and had you tilt your head to the right, we're going to push this mushroom to the right. Your eyes are going to try to keep you static by bouncing to the left. This involuntary eye movement is called nystagmus, and vice versa. If the fluid motion, we'll change it to red, is coming this way, our mushroom will bounce to the left, and again, as that fluid slows, that mushroom is going to kind of wobble its way back into position, at which point our eyes will stop moving, but our eye will undergo involuntary eye movement and bounce to the right. The test that we can use to check for dynamic equilibrium is called Barony's test, and we do this in lab. We can follow our first, second, and third order neurons for equilibrium or balance. We follow from the vestibular branch, first order, into the medulla oblongata. We follow it up to the thalamus. Now at the medulla oblongata, we're talking about the vestibular nuclei located there, and then up second order neuron to the thalamus, and then third order neuron to vestibular area of the cerebral cortex. Now, table 17.2 bundles everything into a nice, neat package, looking at the regions of the ear and their key structures. It highlights it, colors it. This is a good way to kind of break everything down into a nice, neat little note card to help you study. The last thing we're going to talk about is aging and what happens to our special senses. So around the age of 50, smell and taste are affected by aging. The receptors are gradually lost and the regeneration occur at a slower rate. Around the age of 40, when we talk about our eye, our lens begins to lose elasticity and has trouble focusing on close objects. This is why our parents and grandparents need cheaters. So we talk about these glasses that they put on when we're at restaurants to read the menu or to look at their cell phones. Again, these cheaters help them focus on close objects. Why is this happening? Muscles of the iris weaken and react more slowly to light and dark, resulting in difficulty adjusting to changes in lighting. We could look at some diseases. Retinal diseases such as macular disease, detached retina, and glaucoma occur more frequently in the elderly. Remember, we said glaucoma was a buildup of pressure in that anterior chamber, right? So where we're not draining enough aqueous humor into those canals of Schlemm. By about the age 60, approximately 25% of individuals experience a noticeable hearing loss, okay, presbycusis, this is what we talk about, and sometimes we get an increase in ringing in the ears, which is known as tinnitus. So here's tinnitus, ringing in the ears, which is and can cause also vestibular imbalance, which is also going to occur more frequently in the elderly, and we can call this vertigo.